In 1974, a team of researchers at Oxford University sat down with 102 volunteers, and a simple, maddening question that had bothered humans for as long as they'd been swatting at their own arms in the dark. Why do mosquitoes bite some people so much more than others, even when everyone is standing in the exact same backyard at the exact same time, wearing the same amount of exposed skin, breathing the same evening air? The researchers measured skin temperature, body fat percentage, pigmentation, age, sex, diet, and almost as an afterthought, blood type. When they pulled blood from the mosquitoes that had already fed and compared it against their volunteers, one pattern stood out. People with blood type O seemed to get bitten more. That single finding from a single study would go on to shape 50 years of popular belief, spreading from a modest academic paper into magazine, articles, medical journals, casual health websites, and millions of quiet conversations at backyard barbecues, with people carrying type O blood half jokingly, half seriously blaming their own genetics every summer for every fresh welt on their ankles. It felt tidy. It felt provable. And for half a century, almost nobody seriously questioned it. Then scientists actually started sequencing the DNA inside the mosquitoes themselves, and the entire story started falling apart in ways nobody expected, eventually leading somewhere far stranger than blood type ever was. Your skin is covered in trillions of bacteria, and those bacteria, not your blood, appear to be the ones actually calling the mosquitoes in. The blood type theory didn't die quietly, and it didn't die quickly either, holding on with real staying power for decades, even as the evidence supporting it slowly grew shakier. Throughout the 1980s and 1990s, research teams kept trying to confirm the Oxford result, and the results kept coming back messy. Some studies found the same type O pattern. Others found nothing at all. In 2004, a follow-up study looking specifically at blood type alongside something called secretor status, whether a person releases blood type antigens into their sweat and saliva, found that mosquitoes did land on people with type O blood more often. But here's the detail that rarely makes it into the popular retelling. The part that got quietly dropped every time this study got cited in a magazine article or casual conversation. That result was only statistically significant when compared specifically against blood type A, not against the other blood types tested. Already the tidy story had a crack in it, a crack that would only widen with each new study that followed. Then, in 2014, a study published in the peer-reviewed journal Acta Tropica set out to settle the question more rigorously, testing mosquitoes against artificial membrane feeders loaded with different blood types alongside direct exposure to human hands. What they found didn't just complicate the type O theory, it inverted it completely. Mosquitoes fed on blood type AB fully 40% of the time, compared to 24% for type A, 21% for type B, and just 15% for type O, the supposed mosquito magnet blood type. Dead last, precisely the group half a century of popular belief had assured people they should worry about least. 50 years of casual certainty upended by a single carefully controlled experiment, and yet the AB reversal never gained anywhere near the cultural traction the original 1974 finding did. A reminder that the version of a story that spreads widely isn't always the version that turns out to be right. The confusion deepened from there rather than clearing up. Different mosquito species didn't even seem to agree with each other, which should have been the first real hint that something more universal than blood type was actually at play, since blood type itself doesn't change, depending on which species happens to be doing the biting. Some research on Anopheles stephensi, the mosquito responsible for spreading malaria across large parts of Asia, suggested a strong preference for blood type B specifically, with the original study reporting that mosquitoes fed on type B blood even laid substantially more eggs than those fed on other types, framing it as a discovery with real implications for predicting malaria risk. That particular finding briefly became one of the most cited pieces of evidence in the entire field, published in the journal Scientific Reports in December of 2021. Other Anopheles species showed no consistent blood type preference whatsoever. The pattern that was supposed to be universal wasn't even holding steady within a single genus of mosquito. Faced with results that contradicted each other, study after study, decade after decade, scientists eventually asked a more honest question. What if blood type simply wasn't the primary factor at all, and everyone had just been measuring the wrong variable for 50 years running? Answering that required a completely different kind of evidence than anything the original 1974 study could have gathered, because it required looking directly at real mosquitoes that had already bitten real people in the real world, not laboratory volunteers, sitting still under controlled conditions, waiting patiently to be counted. The breakthrough came from DNA sequencing technology that simply didn't exist in any practical, affordable form even a couple of decades ago. Instead of exposing volunteers to mosquitoes in a lab and counting landings, researchers began collecting mosquitoes that had already fed on people in natural settings. then extracting and sequencing the human DNA sitting inside the insect's own digestive systems, effectively turning each fed mosquito into a tiny, unwitting witness that could name exactly who it had bitten. One study focused on mosquitoes, collected from both lowland and highland sites in western Kenya, extracting blood meal DNA and sequencing specific genetic markers that functioned almost like a barcode unique to each person who had been bitten. This approach sidestepped the artificial conditions of a laboratory feeder entirely. It measured what mosquitoes were actually doing in the field to actual people going about their actual lives, sleeping in their own homes, walking to work, sitting outside in the evening without a research assistant watching and counting. And what it found was that blood type, when any correlation showed up at all, explained only a small sliver of the story. The real variation between who got bitten constantly and who barely got touched had almost nothing to do with which blood type sat in their veins. If it wasn't blood type, the obvious next question was what actually was driving the difference, and the answer came from an area of biology most people rarely think about at all: the ecosystem of bacteria living directly on human skin, an invisible population numbering in the trillions on every single person alive. Scientists had long known mosquitoes hunted primarily by smell, tracking exhaled carbon dioxide from meters away, and then honing in on lactic acid and other volatile compounds released from the skin itself. What took much longer to fully appreciate was that many of those specific compounds aren't actually produced by human cells directly. They're metabolic byproducts of the bacteria living on the skin's surface, organisms like Staphylococcus and Corynebacterium species that exist by the trillions on every person's body, quietly processing sweat, oils, and dead skin cells into the exact volatile chemical signatures mosquitoes use to find their next meal 24 hours a day, whether their human host is aware of it or not. A study published in the journal Scientific Reports brought this into sharp focus, quantifying the volatile compounds produced by dozens of distinct skin bacterial strains, and then running behavioral experiments to see which specific volatiles actually pulled mosquitoes in. The conclusion was about as direct as scientific papers ever get. The resident human skin microbiome was found to be responsible for producing most of the compounds that draw mosquitoes toward a given person in the first place, not blood. Not some fixed genetic signature locked into your red blood cells since birth. A living, shifting population of bacteria on the surface of your skin, different in its exact composition from one person to the next, generating a distinct, individualized scent profile that mosquitoes are reading constantly. This single reframing quietly answered nearly every contradiction the blood type research had struggled with for decades. Why did some studies find a real O preference, while others found none at all? Because the actual driver, skin bacterial composition, varies enormously between individuals and even between different testing conditions, while blood type never changes and was only ever weakly correlated with it in the first place. Why did different mosquito species seem to prefer different blood types in different studies? Because different species have evolved to key in on slightly different volatile compounds produced at different concentrations, depending entirely on which bacterial strains happen to dominate a given person's skin, not on their ABO status. Why do identical twins tend to attract mosquitoes at strikingly similar rates, while fraternal twins show far more variation between each other? Because identical twins, sharing essentially the same genome, tend to cultivate closely related skin bacterial communities, producing nearly identical volatile signatures, while fraternal twins, genetically no more alike than typical siblings, develop more divergent bacterial populations and more divergent scents as a result. Researchers dug further and found that the composition of someone's skin bacteria mattered more than sheer bacterial quantity. People with a higher diversity of bacterial species on their skin tended to be noticeably less attractive to mosquitoes overall. People with lower diversity, particularly those whose skin was dominated by high concentrations of Streptococcus species, tended to be significantly more attractive. The mechanism traces back to specific metabolic byproducts. Lactic acid functions as a powerful mosquito attractant on its own, and when it shows up alongside exhaled carbon dioxide, mosquitoes respond to nearby skin volatiles with dramatically heightened sensitivity, essentially priming their entire host-seeking response. Highly attractive individuals tended to host bacteria producing elevated levels of volatile fatty acids and long-chain carboxylic acids, compounds that function almost like a chemical beacon, detectable from a genuine distance. People with more diverse bacterial ecosystems, by contrast, tended to host strains producing short-chain fatty acids instead, compounds that appear to actively suppress mosquito landing behavior rather than encourage it. The skin, it turns out, isn't a passive surface mosquitoes happen to smell. It's a living chemical factory running continuously beneath your notice, and the workers running that factory are bacteria, not blood cells, punching in and out of shifts you never asked them to work. Once scientists understood that bacteria were doing the actual work, an obvious and genuinely exciting question followed almost immediately. If bacterial composition drives mosquito attraction, Could bacterial composition be deliberately engineered to reduce it? In 2024, researchers at the University of California, San Diego, working with Stanford University's Microbiome Therapies Initiative, set out to test exactly. Led by Professor Omar Akbari, the team genetically modified two extremely common human skin bacteria, Staphylococcus epidermidis and Corynebacterium amicalatum, deleting the specific gene responsible for producing L+ lactic acid, the same compound identified as one of the most powerful mosquito attractants on human skin. Staphylococcus epidermidis in particular is considered one of the core, near-universal members of the human skin microbiome, present on almost everybody's skin already, which made it an especially practical target, and one that had reportedly proven notoriously difficult for earlier researchers to genetically modify at all before recent advances made it possible. The results, published in the journal PNAS Nexus, were striking enough to draw attention well beyond the microbiology community, quickly picked up by science journalists, covering it as one of the more genuinely promising developments in mosquito control research in years. Mice carrying the engineered bacteria saw mosquito attraction drop by as much as 64.4%, compared to mice carrying the unmodified strains, a reduction that held for a remarkable 11 consecutive days. Broken down by species, engineered Staphylococcus epidermidis cut attraction from Aedes aegypti, the mosquito responsible for spreading dengue and Zika, by roughly half, produced a similarly steep drop against Anopheles gambiae, a major African malaria vector, and reduced attraction from Culex quinquefasciatus, a common nuisance and disease-carrying species found across much of the world, by about 22%. Even among the reduced number of mosquitoes that still landed, actual biting behavior dropped further still, an effect that lasted for a full 7 days. For comparison, DEET, the chemical repellent that has functioned as the gold standard of mosquito protection for decades, typically provides somewhere between 4 and 8 hours of protection before it needs to be reapplied, meaning a single application of engineered bacteria potentially outlasted it by a factor of more than 30. A living bacterial treatment lasting 11 uninterrupted days, if it can eventually be safely adapted for human skin, would represent an entirely different category of protection. Not a better spray, but a fundamentally different approach that works with the body's own microbial ecosystem, rather than layering a chemical barrier on top of it. None of this means blood type is completely irrelevant, and it would be dishonest to claim 50 years of prior research were worthless, or that every researcher who ever studied blood type was chasing a mirage. Some mosquito species do still show mild statistical leanings towards certain blood types under certain conditions, and those leanings may connect to subtle differences in the specific antigens some people secrete onto their skin alongside their bacterial ecosystem. But those effects are consistently small, inconsistent across species, and dwarfed by the much larger influence of bacterial volatiles. Blood type turned out to be a real but minor variable, trapped for decades, inside a story that gave it far more credit than the evidence ever actually supported. The fuller picture involves plenty of other factors layered on top of the bacterial story too. Metabolic rate plays a measurable role since people who burn energy faster exhale more carbon dioxide with every breath, and carbon dioxide is the very first long-range signal mosquitoes use to detect a potential host at all, sometimes from more than 50 meters away depending on wind conditions. Scattered flint tools on the surface and moved on, never suspecting what lay directly beneath their feet. It took until 1994 for the significance to actually register, when a German archaeologist named Klaus Schmidt, touring the region to catalog Neolithic sites after previously working nearby at Navalichori, recognized what he was looking at and understood, almost immediately, that this was something the field had never seen before. Standing among the scattered surface stones and the worked flint, he reportedly realized within moments that he could either walk away and let someone else eventually stumble onto the discovery of the century, or dedicate the rest of his career to. He chose the latter. Schmidt spent the next 2 decades of his life excavating and studying Göbekli Tepe, and his research fundamentally upended the model archaeologists had relied on for most of the 20th century. That older model was tidy and linear. Nomadic hunter-gatherers eventually developed agriculture. Agriculture allowed permanent settlement produced surplus food allowed specialization, and specialization eventually produced priests, architects, and the monumental religious structures that come with organized society. Agriculture first, monuments later. In other words, a sequence taught in university lecture halls for generations, as though it were simply the natural, inevitable order in which every human society on the planet had to develop. Göbekli Tepe broke that sequence completely, and it did so in a way that left the field with no tidy way to simply patch the old model and move on. Here was a monumental religious complex, built by people with no agriculture, no permanent settlements by the old definition, and no metal tools, who nonetheless organized enough labor to move 50-ton stones and carve them with genuine artistic mastery. Schmidt's interpretation was that mobile hunter-gatherer bands from across the wider region gathered here periodically for shared ritual, a pilgrimage site rather than a home, a theory strongly supported by the more than 100,000 animal bone fragments excavated from the site, evidence of massive communal feasts where visiting groups ate and celebrated together before dispersing again, likely to their own separate scattered territories across the surrounding hills and river valleys. Gazelle bones dominate much of that faunal assemblage, alongside wild cattle, boar, and a wide range of birds, suggesting organized hunting expeditions, timed specifically to supply these gatherings, rather than opportunistic, everyday foraging, a level of logistical coordination across scattered mobile groups that itself argues for a surprisingly sophisticated shared social structure. Under this reading, Göbekli Tepe functioned less like a town and more like a recurring festival ground, a place where otherwise scattered mobile communities came together periodically to reinforce shared identity, trade information and marriage partners, and jointly maintain a sacred site none of them could have built or sustained entirely alone. Klaus Schmidt died in 2014 at 60 years old during an active excavation season, and his death genuinely shook the archaeological community, in part because he never lived to see some of the discoveries that would go on to complicate, and in some ways directly challenge, his own original interpretation of the site. After his death, the excavation continued under Muslim Erkan of the Schanlurfa Museum and Lee Clare of the German Archaeological Institute. And in 2020, the project's overall directorship passed to Professor Nekmi Karul of Istanbul University, with Clare continuing on as coordinator. Under their leadership, Göbekli Tepe has become the centerpiece of something called the Taş Tepeler Project, a coordinated regional effort now spanning 10 related archaeological sites across southeastern Turkey, involving 36 international institutions and more than 200 researchers and students. One of those sister sites, Karahan Tepe, sits close enough to Göbekli Tepe to belong to the same broader Neolithic cultural sphere, and its own T-shaped pillars and carved human and animal figures have repeatedly turned out to mirror discoveries made at Göbekli Tepe itself, each site's findings quietly cross-checking and reinforcing the others, building a much richer regional picture than either location could offer entirely on its own, and suggesting this entire stretch of southeastern Turkey once hosted a genuinely interconnected network of ceremonial sites, rather than a single isolated anomaly. In September 2025, Turkey's Minister of Culture and Tourism, Mehmet Nuri Ersoy, stood at the site alongside Japan's Princess Akiko of Mikasa and announced something genuinely new. This was not a decorative flourish or an accident of construction. Researchers concluded the statue had been placed there on purpose, most likely as a votive offering, and Ersoy noted that similar human figures had previously turned up at the nearby related site of Karahan Tepe, but never before in this specific, deliberate wall placement at Göbekli Tepe itself. It offered, in his words, a deeper window into the ritual life and symbolic world of a Neolithic community archaeologists are still working to fully understand. Ersoy went further than the discovery itself in his remarks that day, describing Göbekli Tepe not merely as a Turkish national treasure, but as, in his words, the shared memory of all humanity, a responsibility to preserve and pass forward rather than simply a tourist attraction to promote. That statue wasn't the only thing complicating the old story. Beginning around 2025, archaeologists working the site started turning up something the classic pilgrimage site theory did not really predict. Grinding mills used for processing grain, flint sickles clearly designed for harvesting, and plant residue consistent with sustained food preparation, alongside what appear to be actual residential structures rather than purely ceremonial spaces. The kind of everyday domestic tools that show up in a place where people are cooking and eating meal after meal rather than passing through once a year for a single shared feast. None of that fits comfortably with a picture of mobile bands passing through occasionally for a shared feast and then moving on. It fits far better with a picture of at least some people living at, or very near the site semi-permanently, possibly year-round, engaged in early informal forms of food cultivation long before agriculture, as we typically define it, had fully emerged elsewhere in the wider region. If that holds up under continued excavation, it flips Schmidt's own sequencing in a genuinely radical direction. Instead of agriculture and settlement coming first and organized religion following afterward as a product of that stability, it raises the real possibility that shared spiritual practice and the pull of a sacred gathering place is what convinced people to stay in one location long enough to start experimenting with cultivation in the first place. Belief, in other words, might have been the thing that built the foundation for farming, not the other way around, a genuinely uncomfortable reversal for a model archaeology had leaned on for the better part of 100 years, and one that continues to generate real, unresolved debate among specialists working the site today, precisely because it would mean rethinking not just this one site, but the standard timeline taught about the origins of civilization more broadly. Now, for the part of this story that sounds almost too strange to be real, and yet comes from a peer-reviewed paper rather than a viral video. In 2024, Professor Martin Sweatman, an engineer in the School of Engineering at the University of Edinburgh, who has spent years applying statistical and symbolic analysis to Ice Age and Neolithic art, published a study in the academic journal Time and Mind titled Representations of Calendars and Time at Göbekli Tepe and Karahan Tepe. Sweatman focused on a specific pillar, Pillar 43, standing inside enclosure D, covered in rows of small V-shaped and square symbols. His analysis found that these symbols, counted carefully across the pillar, add up to 364, arranged in a pattern consistent with 12 lunar months, plus an additional stretch of days, a lunisolar counting system rather than a purely solar one.